Hello, everyone. My name is Jay, and I'm honored to be invited for presenting the results of a forensic investigation that started with a misplaced protein. We're calling the organism responsible Contaminivirus ubique. Our investigation begins while we are analyzing high-value research samples. Specifically, we were reviewing data from a CD8 plus T cell study using non-obese diabetic mouse. Non-obese diabetic mice are the leading animal model for type 1 diabetes research. This study's primary goal was to identify the molecular triggers and pathways of T cell activation to halt autoimmune destruction. However, in the midst of searching for disease-related proteins, we isolate an unusual fragment, a contaminant ubique or C-ubique. It was the core replicase enzyme, the RDRP of a virus belonging to the Totiviridae family. The Totiviridae family, where our new virus cluster is the archetype of simplicity. Their function is minimal. They rely entirely on the host cell, primarily fungi, to create just two core proteins, the shell and the engine, and nothing else. They don't even exit the cell. They rely on host-to-host transmission during cell division or fusion. Currently, the most famous member of this family is the LA virus, the classic model found in baker's yeast. LA virus showed us how these genomes work. The gag or shell and the pole or engine are expressed from a single messenger RNA via highly precise -1 ribosomal frame shift. This complexity is built into their minimal structure. But you might wonder, Totivirus typically infect fungi. Why were they detected in mouse T cell? When we examine sequence taxonomy, a apparent explanation emerged. The CD8 sample contains 8.74 Malassezia, which is a common fungi genus and a known host to Totivirus, which at first glance suggests a plausible biological co-occurrence. However, the experimental context is critical. This data set derived from a highly purified flow-sorted T cell population. At this level of purification, nearly 9% of fungal signal is far beyond acceptable. This level of fungal signal becomes the key warning sign. This inconsistency leads to our core hypothesis. The detection of C-ubique in non-fungal sequencing libraries reflecting systematic contamination that tracks with a global sequence reagent supply chain rather than true biological infections. To test this hypothesis, we move beyond the individual samples and initiate a global cross-project investigation of 154 bioprojects as shown in the map. Within the 154 project, we handpicked 16 diverse sequence projects, from mouse model to human RNA-seq, and even global B viral. Our goal was to see if the virus tracked with biology or with library reagents. To move beyond correlation and establish definitive evidence, we are also looking to characterize the organism by reconstructing the viral genome from scattered genomic fragments. The overall goal is to rebuild the viral body using a structured three-step reconstruction strategy as shown in the slides. Together, this workflow allows us to move from an isolated ambiguous sequence fragment to a fully reconstructed and functional annotated viral genome. Moving to the result. The genome reconstruction is successful, but reveals an unexpected complications. Functional annotation using inter-proscan provided unambiguous results. Segment 2 encodes a complete RNA-dependent RNA polymerase, while segment 1 encodes the structural shell component. But the original C-ubique couldn't map to the reconstructed RDRP opening reading frame. This result indicates that the contamination is more complex than initially assumed. We next play this finding in context by examining the 16 project level association identified in the global audit. To do this, we perform a T-blast N search using the C-ubique RDRP fragments against all available SRA datasets, assessing how many shared high sequence identity. The results are unambiguous and resolve into two distinct non-overlapping clusters. The first cluster represents a native lineage, This sequence are consistent with natural occurring environmental virus and displaying this substantial sequence divergence ranging from 70 to 96% identity. The second cluster corresponds to the contaminant lineage, which includes ubique. In this group, segments from laboratory worldwide, independent of geographical locations, host organisms, or sequence methodology, classed into one single high conserved cluster with 98 to 100% identity. This uniform global 100% identity is the abiotic signature. It proves the sequence is a static industrial product, not an involving biological entity in the sample environment. The key observation is the presence of an exact 100% identical match in Project 14, which have zero detectable fungi. This is the critical evidence. It breaks the biological co-infection model. The virus is present in a pure sequencing environment without a fungal host to explain its origin. This finding confirms that a 100% identity is the definitive abiotic signature. It proves that this sequence is not an evolving biological entity, but a static molecular clone that has successfully colonized the global reagent supply chain. Characterizing the ubique sequence provide the critical insight that viral contamination is not a sporadic noise, but a systematic, highly stable biological artifact that requires standardized filter across all sequence disciplines. These emphasize that Totiviridae, known for their simple structure and persistence, are unique successfully in contaminating the reagent supply chain, pushing against the textbook definition of a virus that must be dependent on a living host for spread. This brought us to the final takeaway. First, members of the Totiviridae family are uniquely successful at contaminating the sequence reagent supply chain. Their simple genome organization, exceptional environmental stability enables a single molecular clone to persist and spread throughout global laboratory infrastructure. Secondly, C-ubique propagates through ostensibly a sterile reagent and buffer, challenging the conventional definition of virus as an entity strictly dependent on a living host for transmission. In this context, the sequence effectively bypasses biological hosts entirely and instead exploit laboratory workflows as its primary distribution mechanism. Given its prevalence and stability, the abundance of C-ubique should be adopted as a mandatory quality control metric across all sequencing disciplines. To protect integrity of RNA-seq and transcriptomic data, standardized, filtered, and reporting based on this known contamination must be incorporated into routine and analytic pipelines. Thank you, and I'm welcome for questions.